talking to a group of intelligent people from different backgrounds is really, I find it among the most difficult talks I give because you've all got different backgrounds. There's a lot to talk about. So forgive me if I miss the mark, but we'll do what we can and to cover different areas of genetics of blood cancer. So hopefully at least give a framework, and hopefully I'll leave some time for questions at the end. Half of my life I spent as a diagnostic haematologist, so looking at your samples down the microscope and making diagnoses, and that's where the focus will be, a tiny bit on our research at the end. Genetics it's a big word, isn't it? We use the word genetics in so many different ways, and so I want to sort of think about all the words that we use. We talk about genes, genetics, genomics, and so on. Often when we think about genetics, we think about familial things when we're talking in the, out in the real world. So we talk about stuff you inherit from your parents, the heritable genes. The germline, is that a word you use? The germline, the stuff that you pass on to your kids with the sperm and your eggs, that's the germline. And so we think about genetics of the germline, or our constitutional genetics, the stuff that makes us what we are as a person. But then from a cancer perspective, most of what we're interested in is the genes within the cells. So we think about cancer genes or mutations of those cancer genes in the cells of your bone marrow as opposed to the rest of your body. And so we call it somatic. The word somatic's not a general word we use out on the street. Psychosomatic, you know that word with the control of the brain over the body. Somatic is about the body's genes as opposed to what you were born with. So we talk about somatic changes or somatic genetics, and so much of blood cancer's around that. And then we've got this expression, the genome. So every cell has got all our genes packed in our chromosomes. So we talk about our genome, which is all of our genes and DNA all together. So many different uses of the word genetics and genes. So it does get a bit complicated. Now, just some background information. When we were conceived, we got a bunch of genes from mum and a bunch from dad. They're all packaged in chromosomes. So we got 23 chromosomes from mum and 23 chromosomes from dad. In those chromosomes are packed all the letters of DNA that I'll show you in a bit. So we've got 3 billion letters of DNA from mum and 3 billion from dad. So we've got in every cell in our body, we've got 6 billion letters of DNA. And we can look at the chromosomes, and so this is sort of our first approach really to genetics, is to look at the chromosomes, and it's called a karyotype. We can see in the middle there is a cell that's been a special stage of just about to divide, splattered onto a slide, and you can see them there, and you can cut them out with scissors or a computer and line them all up and see our 46 chromosomes of a female there, 2X chromosomes. So that's a technique we use a lot that's really important in a lot of areas, especially myeloma. Now, chromosomes, I think of them like recipe books. We've got 46 recipe books, 23 from mum, 23 from dad. And the chromosomes contain genes. We've got about 20,000 genes that makes us what we are. So genes make all sorts of things, like genes make hemoglobin. There's several genes for hemoglobin. Genes have the recipe for insulin, for example. The genes have recipes for things that cause cells to grow and divide, and they're genes that tell cells to stop growing and dividing, and so of course they're important to us. So I like to think of them as like recipe books. When the human genome, this is all the letters of the DNA, were first decoded about 20 years ago, the guys at Wellcome published them in a series of books there. So they put out all those volumes, and it's really dense text in there with all the 6 billion, or 3 billion from each side, letters of the genome. And you can look up close. It's just a series of A, C, T's and G's, which give us a code for our genes. This is what we carry around from cell to cell. Now, I like to think of these like recipes. So I was looking at Nigella's recipe and pulled out her recipe for lemon and almond cake. Take the zest of 2 lemons, some butter, some sugar and eggs. And There's the recipe for hemoglobin, just the beginning of the recipe for hemoglobin. So we take a few of those letters in the right place for hemoglobin, and it starts with ATG, which means take a methionine. And then the next one, GTG, add a valine, add a histine, add a leucine, add a tyrosine, proline, glutamate, another glutamate. And you build up all those little building blocks, amino acids, to make the protein that we call hemoglobin. So it's just a series of recipes like that. Now, the other thing to think about, we start off with one cell at the beginning of life, one cell, and then we end up with 37 trillion cells in our body. Now, some clever person has counted them. What's really cool is that 26 trillion of them are blood cells. So it just shows how important hematology is. So about 2/3 of all the cells in our body are blood cells. But to get from one cell to 37 trillion cells, we've got a lot of dividing to do. The cells have got to divide and divide and divide all the way through, making and renewing new cells all the time as well. So in addition to getting there, we're making new ones every day. So every day we make about 250 billion new red blood cells and 30 billion new white cells. The problem is we make mistakes. And this is largely why you're in the room, is because there are mistakes occur in this process. I want to talk a little bit about mistakes because I think it's really important. I want to talk about some work that Peter Campbell's done. Now, Peter Campbell's a Dunedin boy. His father was the dean of the medical school, John Campbell, and Peter's been in England at Sanger Institute for a long time now. He's one of UK's famous scientists. He's one of our best exports. And he's been doing some absolutely amazing work, and he's a haematologist in blood cancer. But what they've done recently, it's really quite recent data, is they've actually quantified how many mistakes our blood cells make through life. And so every time a stem cell, you know, all our blood comes from stem cells in the bone marrow, and all our cancers come from stem cells. Every time a stem cell divides and reproduces itself, it's got to reproduce 6 billion letters of DNA, and it makes mistakes. And he's quantified that, and he says that every year, every single stem cell in our body accumulates 17 mistakes, 17 mutations. And incredibly linear here, you can see on the plot that this is there's thousands of stem cells from many individuals, and that's an incredibly linear approach, that it's very consistent. So that by the time you get to my age, a bit over 60, then every one of my stem cells has made 1,000 different mistakes. So each one makes its own mistakes. Now, given that we've got about 100,000 stem cells, and everyone's making, by our age, 1,000 mistakes, it's not surprising that sooner or later something goes wrong and we end up with a blood cancer. Now, I suppose what is surprising, most of the time we don't, and most of the cells don't end up with blood cancer. Part of that, these things I call mutations or mistakes from the middle of nowhere. They don't affect anything important, they don't affect important genes, they're just sort of random errors that just are not on the radar in terms of how a cell functions. But it's just the truth that we just make mistakes every time a cell divides. So these we call acquired mutations, or in our jargon, somatic mutations. They're happening in the body. They're not from your germ cells. They're not you as a whole. You're not going to pass them on to other people. They're in our bone marrow, and our bone marrow stem cells. Now, these mistakes can be just a single spelling error, so a point mutation. I'll show you an example of that. Sometimes you delete a little bit of DNA or insert a little bit of DNA, and a good example of that would be the FLT3 internal tandem duplication that's seen in quite common in acute myeloid leukemia. And a big theme, though, are translocations, which is when chromosomes cross over. So I'm going to talk quite a bit about translocations. They're quite important in myeloma particularly. Well, they're important in all of the blood cancers. So each of those has different importance in different places. This is just a point mutation here. So let's say that the Nigella said zest and juice of 2 demons instead of lemons, and that would be a bad cake. And the same thing happens here in this gene called JAK2. Now, if there's people in here with myeloproliferative neoplasms, JAK2 mutations, very, very common. And there's just one spelling error. So somewhere in your life, maybe decades ago, a G changed to a T. Just one spelling error out of 6 billion. And so instead of taking a tyrosine glycine valine, that valine there got changed to a phenylalanine. And that changed the whole way that this gene, this protein called JAK2 works. So that's pretty common in polycythemia vera, and almost everyone with polycythemia vera has that. Essential thrombocythemia and myelofibrosis is pretty common. Just that one single point mutation in the wrong place, if you like to think of it. I like JAK2, just a little bit about JAK2. I like to think of JAK2 as being like the inside of a light switch. It's not the outside of a light switch. We don't have light switches here. It's not the outside, but you switch. It's on the inside of the light switch. So imagine a light switch where the inside was always turned on and you just flick the switch and it made no difference. It was always turned on. So JAK2's like that. It's when it's the inside of the switch is always turned on. So a normal JAK2, the centre there, that man is making red cells, turning on the switch on and off. But if a mutated JAK2, the red cells are just coming out of that machine or whatever he's making, all by themselves, out of control. So that's what happens with JAK2, that single point mutation. I want to talk a bit about translocations now. Every time a cell divides, you've got all these chromosomes, 46 chromosomes, and you've got to make another cell. So you've got 46 chromosomes in your beginning cell, now you've got 46 plus 46. And those chromosomes, which are in blue in the middle, are pulled apart by the green stuff and separated into 2 new cells. Now, chromosomes are a bit like spaghetti, and it's not surprising that sometimes they get tangled up in that process. What is surprising is that the vast majority of the time they don't, but sometimes they do. And so we have these translocations. So an example here on this one, I've got an image of chromosome number 4 and chromosome number 20. And do you want the stool? And for some reason, during a particular cell division, somewhere in the middle of those 100,000 stem cells, those two got tangled up, and part of chromosome 4 went on to chromosome 20, and part of chromosome 20 went on to chromosome 4. They just sort of got tangled up and headed off in the opposite direction. And so we call that a 420 translocation. T for translocation, 420. So the famous translocation here, the most famous of the translocations is it's called the Philadelphia chromosome, which is involved in chronic myeloid leukemia. It was called Philadelphia chromosomes because it was discovered in Philadelphia. It was the first essentially the first cancer genetic abnormality or cancer chromosome to be discovered. So it was put hematology on the map a long time ago. And the fascinating thing there is you take chromosome 9, chromosome 22, there's a couple of genes there. They cross over and one gene gets fused onto another gene. And at the bottom, it makes a brand new gene. Half of one gene, half of another gene makes a brand new gene. So people with chronic myeloid leukemia are really special because they've got a gene that nobody else has got. The problem is that gene makes a protein. Genes make proteins. And the protein does weird stuff to the cells and makes the cells grow. And that's chronic myeloid leukemia, just on a single translocation causes it. The great news about chronic myeloid leukemia is that particular special gene, special protein that they have is very susceptible to inhibition by drugs like imatinib and the imatinib-related drugs, which are very, very successful. So that's a really nice story because that molecular target, was targeted very successfully. Translocations occur all over the place. So on this diagram, there's lots of translocations in the blood cancers, but there's translocations involving other cancers or even schizophrenia there. I'm not aware of that, but a wide variety of things result from translocations in a lot of different chromosomes. Some of the ones that we think about a lot, I've got some myeloma ones on the left. 11, 14, 4, 14, 14, 20. We're going to come back to 14 in a minute. When I'm trying to diagnose mantle cell lymphoma, it can be a tricky diagnosis, and I really like to see the 11-14 translocation. Got the 11-14? Probably, yep.
No, not TP53. So.
Right, cool. But it's one of our diagnostic markers, which if there's any uncertainty, we'll do that. Follicular lymphoma is usually straightforward, but it's got a 14-18 translocation. Acute lymphoblastic leukemia has got its own set of translocations, and acute myeloid leukemia has got common translocations. So they're all over the place, and they provide really important diagnostic, prognostic, or treatment-related information. Now, I'll come back to translocations in a minute. Somewhere in the bone marrow, one of these mistakes occurs. So we've got a point mutation, perhaps, we've got a translocation, a deletion, something like that. It happens in one cell in the bone marrow, so I've got a green cancer-initiating cell. This is not in the rest of your body, just in your bone marrow. And for some reason, that cell thinks it's special. It maybe divides a little bit more quickly, or maybe it doesn't die when it should die. And that over many years, many decades, maybe in some cases, from some of going back to Peter Campbell's work, maybe over most of your lifetime, this can occur. That cell slowly divides and divides and divides and becomes more and more prominent. And then additional mutations often happen. And so I've got the red, it's not my diagram, someone else's diagram, the red is additional mutations, and that cell becomes more and more successful. So it's like evolution, it's like survival of the fittest, and so that clone of cells grows and grows and grows and starts to mess with our bodies, doesn't it? Gets in the way, crowds out the bone marrow, or causes a lump, or causes damage to your bones. I want to go through some examples using acute myeloid leukemia. Now, a lot of these slides, the next few slides I've got from the Leukemia Blood Cancer Research Group in Auckland. This is Stefan Bollender and Peter Browett and others. And so I borrowed their slides. So acute myeloid leukemia, I looked down the microscope and I said, well, that's acute myeloid leukemia. But we need to know a lot more than that to determine which treatment is the best and get some prediction of how the patient's going to do. To start with, we look at the chromosomes. I'm in awe of the people that look at chromosomes, the cytogeneticists. They're amazing. They can tell these all apart. They know them. It's like looking at sheep. They all look the same to me, but to them or to a farmer, they're all different. And so they look at them and say that chromosome 8 is a bit short and that chromosome 21 is a bit long, and that's an 821 translocation. I just don't know quite how they do it, but they're very good. So an acute myeloid leukemia with an 821 defines a certain subtype that's useful to understand about. Sometimes we can look at them in a bit more detail using FISH. So FISH is where we have fluorescent probes. So on the left-hand side, we've got a green fluorescent marker that targets one little area on chromosome 8, and a red one that targets one little paint, sort of put a paint on one area of chromosome 21. And then you can see right on the far left, you've got places where there's an 8 and a 21, the red and the green, side by side. So they've come together, and that shows the position of the translocation. Now, you don't need to see the chromosomes, so when you look at cells on the right-hand side that are not dividing, but you can see in both of those cells, there's a red and a green side by side, and they sort of go that yellow colour, yellow here. So you can see the cells have all got that 821 translocation. So we use, to analyse the chromosomes in more detail, we often use FISH, especially in myeloma. So once we get the chromosomes, sometimes with some help from FISH, we learn things like the 15-17 translocation is critically important in acute myeloid leukemia. It indicates an acute promyelocytic leukemia, which has very different treatment. So that's critical, and that's one of the ones we get as fast as we possibly can. 922, we've talked about that. It's a Philadelphia chromosome, so that's critical for diagnosis. 1114 is really good to diagnose mantle cell lymphoma, and then we've got myeloma one. So 1114 indicates a myeloma that's going to respond really well to venetoclax. Not that that's available currently, but might be. And then there's other translocations in myeloma, the 414, 1416, 1420, that indicate maybe a myeloma that's going to behave not so well, a higher risk. Possibly, as you said before, if you take a group of people and put them all together, you can say on average, something's going to put you in a higher risk area and something's going to put you in a lower risk area. But if you take each individual by yourself, it's not very predictive, is it? The tools are quite crude. But it's the best we can do to say we should probably have more aggressive treatment, or we don't need to have such aggressive treatment based on these sort of indicators. 14s are quite interesting. Why have you saw in the myeloma, we've got lots of chromosome 14. What's going on there? So we use the example of the translocation 1416. Right, what's a cancer cell in myeloma? Back to school. Plasma cell. Right? Cool. What do plasma cells do for a living? Who's just been at Philip's talk? Plasma cells. Well, that's a good point. They do. But they make antibodies. They make antibodies, immunoglobulins. They make antibodies, don't they? If you had to pick which part of the factory, so the plasma cell's a factory, which part of the factory, which gene is the busiest gene in a plasma cell? That's sort of the antibody gene. The antibody gene's a really busy place. So there's a lot of activity going on around chromosome 14 and the antibody gene. Now, let's say you have a mistake and a little bit of chromosome 16 gets translocated onto 14 and you end up with a 14-16 translocation. What does that do? Well, that brings this gene called MAF. Now, I don't know much about MAF, and I don't expect you to know much about MAF, but MAF gets translocated next to this busy factory. And all that busyness carries over to MAF, and so MAF now gets produced at high rates from the factory. All these workers saying, let's make some more MAF while we're making antibodies. And MAF accelerates cell growth. So that translocation leads to a change which causes the cell to grow faster, which makes the myeloma worse, of course. Now, the translocations help define how someone's going to do. Not absolute, but helps. So these are some examples of translocations in acute myeloid leukemia. So you can see at the top if you've got an inversion of 16, so there's a translocation within 16 itself, or the 15-17. That's pretty good. So we're looking at 75-80% over 10 years. But if you look in some of the other translocations towards the bottom, then the patients don't do so well. So there's a really good starting point to say, hey, we should do really aggressive treatment if you're a young person. Well, maybe if you're older, maybe it's just like, maybe treatment's the wrong thing to do, and maybe we're just going to cause more harm. So that information feeds into people like Philip, who's doing the treating, to make decisions. Now, so we've done the cytogenetics, the karyotypes, the chromosomes, but then underneath this iceberg is all about the genes and the genetics. How are we doing doing all right? So here's Stefan in the Leukemia Blood Cancer Research Unit in Auckland, and what we're still so excited about is this genomic revolution that we're in. So when I started doing research, gosh, if I could do 600 bases of DNA sequence a day, I'd be pretty excited. 300 was good. Now, with his instruments that he's got in the lab, this is old data, so that's 120 billion letters of DNA he can sequence in a day. So that's 200 million-fold increase in ability to sequence DNA. So it's just been an incredible revolution for all of us diagnostically, and that's had impact to you. I keep showing this. This is the cost of sequencing one genome. So to take your cells and say, what does your cancer have in it? Give us the entire readout of the letters of your genome. Now, the first human genome was sequenced in full in 2003, 21 years ago. It cost $3 billion for that first one. And the price has just plummeted. Moore's law is where the price halves every year. It's sort of like Moore's law of computer chips or something, but it's a halving of price every year. And this is on a log scale, so it's not a linear scale, so each line is a 10-fold reduction. And so you can see now the price of sequencing your entire DNA from your cancer cells is less than $1,000, which is just extraordinary. So that's been a massive revolution. As a result, we've discovered lots and lots of genes that are mutated in the cancers. And this is particularly important in acute myeloid leukemias. It's less impact on the lymphomas, myelomas in terms of what we do, but some impact. And so here's a publication that shows the top 36 genes that are mutated in acute myeloid leukemia. So there's lots of them. Now, this has been translated into the sort of panels that are offered. So this is a panel that's offered at Auckland at the research centre. So Auckland-based patients with acute leukemia and myelodysplastic syndrome will be having this panel of genes that will provide information. It's complicated. Now, it's complicated for you, it's complicated for us, but it's very exciting. As a haematologist, I thought by now in my career I'd be starting to get bored, and it's just the exact opposite because of the changes that are occurring here, and it's challenging and complicated. So this is one publication where they've got 150 patients. So the red rectangle there is one patient, the blue one is another patient, and they've lined them all up. And I don't think you could find two patients that are the same on this. They've all got a different spectrum of mutations. So the first one, that patient's got a mutation in TET2, DNMT3A, SRF2, RUNX1, FLT3, ITD, so on. This one's got other mutations. Incredible diversity, which is amazing opportunity and also a challenge for us. Those mutations are feeding into lots of different bits of information. Prognosis, for example. So if you have the one at the top, mutations in CEBPA and you've got leukemia, you do really well, compared to if you've got mutations in TP53, where you do quite poorly. So some really good prognostic information, and some of those affect treatment. So this is, again, a slide from the Auckland group. They did a study once they set it up, and they said, how is it affecting, how are these mutation detections affecting what we do? So they looked at how it refines diagnosis, how it refines prognosis, does it identify targets that I can use to treat the leukemia that I might not otherwise? Can we use it to monitor the disease after treatment and picking up really small amounts of disease after treatment? And what about running in the family? So we're going to come back to this slide later on. These are the sort of results we're getting. We actually, we don't use the Auckland service, they're really busy. We use Melbourne, Peter Mac, and there's a couple of results. So every time we get an acute leukemia, they go there. So patient number one's got a mutation in DNMT3A and IDH2. And IDH2 is interesting because we've got drugs that can work against that. Now, I'm not sure, are they available in New Zealand? I don't think they are yet, are they? Or maybe experimentally, but they're pretty close. So that provides some hope for that patient over and above the treatment that's available. The next patient, quite a complicated patient, they've got 5 different mutations that all add up, and they help us make a diagnosis, to be confident, and also to provide a bit of prognostic information about how they might be doing. So it looks complicated, it is complicated, but it's also very exciting at the same time. Here's an example. Those people with mutations in FLT3, which is a gene, or IDH1 or IDH2, they're just gene names, you don't need to know what they are. There's some really good drugs that are making a big difference in those. So on the left we've got relapsed disease, and on the right complete remission in many of the patients who are treated with those drugs. So the mutations are guiding therapy. The other thing is by knowing the mutations, you can find the leukemia after treatment and know how much is left. Now, when I look down the microscope, if I see less than 5% blasts or leukemia cells, I'm sort of saying, oh, I'm not sure if that's a leukemia cell or just a normal residual cell. Sort of 5% is my threshold. But 5% is a lot of cells, and so 5% of a leukemia cells in the body of someone with leukemia is still 100 billion cells. But if you know what the mutated gene is, we can use these really clever molecular techniques that we have now to go looking for them, and you can pick up one mutated cell in 1,000, 10,000, 100,000, pretty straightforward, maybe in 1,000,000 if you get the right techniques. So it's looking for minimal residual disease or measurable residual disease. So after treatment, looking to see how many cells are there. Is that low enough you can say we're comfortable, or is that high enough that we need to change the treatment? And so that's used a lot in the acute leukemias, chronic myeloid leukemia. Not used routinely in myeloma yet, but it's coming, I think. So that's really useful. So just a bit of a summary of how the panel helped in the acute myeloid leukemia. They changed the diagnosis in 9% of cases. Thank you. Changed the prognosis, or the expected prognosis, in about a third. Identified targets that might be useful for therapy in about a quarter. Allowed markers to measure the residual disease in about 60%. And the other side, which I want to talk about a little bit, is identified mutations that might run in families. And they found more, and the world is finding more than we expect. So I want to talk about that, familial mutations. So I want to emphasise that the vast majority of blood cancers arise somatically in your bone marrow. You don't pass them on to your children. But we are recognising that sometimes they are because of things that you've inherited from your parents or might pass on to your children. Now, myelomas, estimated about 1 in 200 myelomas are familial. Depends on how you define that. Lymphoma, about 1 in 50. There's a close relative who's got the same lymphoma. With acute myeloid leukemia, myelodysplastic syndrome, it's looking at around 5%, and that's been a big surprise recently. And maybe the myeloproliferative neoplasms, that's your polycythemia vera, central thrombocythemia, myelofibrosis, maybe 7 to 8% got familial clustering. We're starting to understand those genes, and I suppose the big surprise from our sequencing is a gene called DDX41. Now, DDX41 turns out to be quite a common, or mutation, a mutation in DDX41, quite a common familial predisposition to leukemia and myelodysplastic syndrome. The curious thing is that the people who get this mutation get their myelodysplastic syndrome or leukemia at an average age the same as everybody else who gets myelodysplastic syndrome or acute leukemia, about 67. So it never really showed up, because we sort of expect people who have things that run in the family to have leukemia at a young age. That's the most common. The one I've got in red is the one that we're seeing most commonly. The others, there's a whole bunch of different genetic mutations affecting those genes that, if they run in the families, will cause generally an early onset of leukemia or myelodysplastic syndrome. Now, in the the right-hand column, I've got penetrance, and it's an expression we use to say, if you inherit that mutation, how often will you get the malignancy? How often will you get the cancer? And so if it's DDX41, only half of people who inherit the mutation will actually end up getting a blood cancer. And say Ankranadi 26, we've got a few families that we've identified around New Zealand, but it's only 10% for an individual that they might get a cancer later on. We're not sure how to deal with those, but it is the subject of a new New Zealand consortium that are trying to deal with this issue. So the issue of what we call penetrance, say you've got a family, you've got 5 members of the family. If it's got variable penetrance, 3 members of the family might be perfectly healthy, but 2 members of the family might have blood cancer. So in that particular case, 40% of the family had the blood cancer. The penetrance is 40%. and they would go on to get treatment. So they're becoming a tricky group, and they're a minority, but they might be affecting some of you. Now, I've got a couple of minutes, haven't I? 10 minutes.
Finishing questions.
And questions.
Yes.
All right. So really quickly then, so most cancers are not related to inherited genes, but we all bring a background with us, don't we? So some of us got paler skin than others, and that's got pluses and minuses, doesn't it? So if we've got pale skin, our vitamin D levels are higher, but we're going to have a higher rate of skin cancer. And same with blood cancer, or blood cell genes, we've got pluses and minuses. So say for lymphoma or myeloma, we might have a stronger immune system and make more lymphocytes and plasma cells, which is great on the one hand, but maybe that makes us ever so slightly more susceptible to those lymphoid cancers, the immune cell cancers. So these are quite subtle, aren't they? It's like being tall or being short or pale or dark or whatever. They're quite subtle. Now, just in the last couple of slides, we can measure these with a technique called genome-wide association studies. So this is a brand new study on myeloma, and they combined the data from 11,000 patients with myeloma, huge numbers, compared with 360,000 people without myeloma. And they profiled across the entire DNA, so they're measuring about nearly 1 million different variants across all our DNA and said, spot the difference. And you can see that in red, pink and red, there are about 27 different genes that contribute to the risk of myeloma. They're all very small. They're all very small. I'm focusing on this one, TFRSF13B, which is one we're particularly interested in. It's called TACI. TACI's easy to say, isn't it? So TACI's a protein that's a receptor on B cells, so it's really important for B cell signaling. And if you have a particular variant of TACI, which is really quite common, it increases the risk of myeloma by about 30%. Now that sounds like a lot, but it's not really, because lifetime risk of myeloma is about 1%. 1% of all people are going to get myeloma, which has surprised me. So it would take from 1% to 1.3%, which is still a low risk, but in terms of understanding it, really important. So we're working on this. Mara and Pacifica have a high rate of this risk variant, and they have a high rate of myeloma. So we've got a study going on at the moment to see if their different genetic background, which has probably given them a stronger immune system, might in fact increase their risk of myeloma. And I'm really excited at the moment. We can talk about it later on, but I've got a student who's working away trying to understand why it gives an increased risk of myeloma. And we've just got some really nice data that suggests we understand what this particular genetic variant does that will increase the risk of cells to myeloma. So it's starting to get a bit technical. So we've got all these different levels of genetics. I'm going to stop. Time for questions, I hope. The microphone, you've got to have the microphone apparently before you ask a question. That's the rule. It's for the recording.
Okay. My question is a statement that I'd like you to talk to. Lots of people get cancers, we then treat them, and then they end up dying of something like AML. So in your presentation, does the chemotherapy cause a gene mutation that then arises at a later date?
Definitely, yeah. So the sort of the classic situation with someone with, say, breast cancer,
yes.
and they get nasty poisons to hopefully cure their breast cancer, but they're poisons and they damage DNA. And so you can get secondary leukemias after the treatment for, say, breast cancer or some other solid tumours. And it's the unfortunate price you pay for that, hopefully that cure. So it's real, and real efforts to try and reduce the toxicity of those therapies and only give as much as you need. So, say, breast cancer, childhood leukemia is another good example. Keep them as low as possible, keeping the survival as high as possible, but minimising the side effects. So definitely it damages the DNA. It's a poison, as you know.
My question is about the geographic distributions. You were talking about Pacifica versus Pakeha. Has there been much work done on Africans and Asians? Is there enough data to make any conclusions then?
There's been work on myeloma on Africans versus Asians, and with, had that fancy picture there, very complicated picture. Well, they've done the same picture for Africans. There are a lot of those risk variants. Variants in DNA that increase the risk. A lot of those are in common with Africans, not all of them. So it can go both ways, because we've all got quite different genetic backgrounds and different mixes of variation, don't we, in different populations. So Bob each way on that one.
Hi, you spoke about gene panels that have been developed by the labs. Would you recommend gene panels to be run for families so we can check for our families?
Oh, that's a tricky one. If someone has, say, an acute leukemia and you run a gene panel and you find a genetic variant that's likely to be germline, so inherited. We've got one at the moment. We've got a lady with a GATA2 variant. And we don't know if it's a leukemia or her body, because it's right on 50%. It's exactly what you'd have if you inherited it. So she's getting hair taken out, and so we're looking at her hair cells to see if she's got it in her constitutional body or whether it's just leukemia specific. If we confirm that she was born with it, inherited, then we would be testing the rest of the family. Or would generally we would offer that testing to the family. So DDX41 is a nice example. We would offer that testing to the others in the family. That becomes important for the leukemias when they go to transplant. So if you're going to transplant a leukemic patient, it's really important to know they didn't get the leukemia because of something they inherited. And it's important to know that when you transplant them with a sibling, if you've got a sibling donor, brother or sister donor, it's really important to know that you're transplanting bone marrow that doesn't have that variant in it. So yes, when we detect them, they're uncommon, we chase them. How far you go with that, we do it for the strong variants, but not the weak variants.
Do you know if there are any genetic studies of blood cancer patients who have... I have blood cancer, my sister has a blood cancer, but my parents had 2 other cancers, not blood cancers, and I'm actually on my 5th cancer.
Right, yeah.
Are there any studies you're aware of?
The Scandinavians, who've got great registries, have done some really good work on this in the past. I haven't sort of followed it, but they've said, if you've got this cancer, what's the chance of having this cancer? And so they did these huge matrices, If you had myeloma, what's the chance that your first-degree relative's got lung cancer, and so on, all those things. And the work has been done. I know, for example, that if someone had lymphoma, their relative was more likely to have lymphoma, or myeloma and lymphoma would go together, but there were lots of other associations. So if I can point you to those, if you come and ask me and give me an email address or something, I can give you that information, but I can't tell you off the top of my head. Yeah.
This question's related, I think. My wife's got multiple myeloma. Her grandfather was subject to mustard gas poisoning in the First World War. That contained melphalan. Yep. Melphalan's used as a treatment in a stem cell. I just wondered if there was any research tying the intergenerational aspects of that.
Can you help me on that? I would suspect, the simple answer is probably not related, because very little of myeloma goes through is hereditary, is familial, very little. I'm not aware of particular research on the mustard gas. There's been plenty of research on atomic exposure of veterans in the atomic testing in the Pacific, and again, there's they didn't really show much that was convincing, but it's really quite difficult to do. Yeah, interesting. Yeah, it's a good question.
I had only one brother. He had CLL. I have multiple myeloma. Is there a connection
Yeah,
there?
maybe. It's hard to know. It's hard to know, isn't it? It could be there's some data to suggest that you're more likely to get that because you've both got these B lymphocyte cancers. But they're both reasonably common, so it could be a coincidence. We can't answer that question, and we don't have the genetic tools yet to be able to dissect. Let's say we could have you and your brother's DNA. We don't yet have the tools to answer that question. So maybe not. Yeah, it's plausible.
To bring it quite recent with COVID, has there been any, probably not because it's recent and studies take time, but has there been any research in how COVID affects the body, affects the immune system, and how that could relate to blood cancers and stuff?
I like that question. Yeah, you do wonder about things that stimulate the immune system and what effect that might have. I'm not aware of anything that will answer that. Are you aware of anything that will answer that? It's too soon, isn't it? What we do, let me say, what we do know about myeloma, for example, is that someone did some really good studies and said if someone has myeloma, when did it start? And their estimate was about 40 years before diagnosis. The beginning of that first cell that thought it was special was probably about, on average, about 40 years earlier. So it wasn't the COVID that you got a couple of years ago. Do you have a follow-up question there?
It's a slightly different topic, but my dad has a histiocyte-based cancer at Chester's, and after he did have COVID, he did a lot of plasma donations. Since it's a plasma-based cancer, would that have affected any of the research, like where that plasma went? Do you think that could have affected or changed anything, given how we're not sure when the mutation started for his cancer?
Would it affect the recipients?
The recipients as well as the research.
Plasma's good because it doesn't contain any DNA. So there's no cells in there, no DNA, so you can't pass on anything with your plasma. So it's safe for a cancer patient to give plasma. Not that we would recommend it, but it's safe. So it's safe for the recipients. I can't really answer your other question about the research. We'd have to sort of think about that offline, and I'll try and answer that. Yeah. One more question over here. Yeah, how are we doing?
Back to the relation between blood cancers. So Scott's got multiple myeloma, but his mother had lymphoma. You were saying that there might be a relation between those
I think
two
so.
cancers?
I think the Scandinavian papers, if I remember them correctly, showed that there was an increase. If you had a family member, first-degree relative with lymphoma, then not only the lymphomas increased, but the myelomas a little bit, if I remember that correctly. So it's just some people are taller, some people are shorter. It's that sort of type of influence, I think, if you know what I mean, rather than the very strong genetic influence. People have looked really, really hard for the familial myeloma genes. Is there a gene, a particular mutation that you've inherited that will cause myeloma? And there's been a lot of work done on that over a long time, and there's really been no success in that. So it's probably sort of a combination of factors, a combination of different susceptibilities that increase our risk because we've got a better immune system, or decrease our risk because we've got a weaker immune system to look. It's a very simplistic way of looking at it, but many, many factors together. A lot of good questions. Where can I write it on? if we go back to the beginning, you got my name. You got my name with one R, right? Yes. Ian.Morison, so one R in Morison, at otago.ac.nz. Or at Awanui Labs, but it's Otago shorter. Yeah. Ian.Morison at otago.ac.nz. Feel free to email me. Yeah, I like questions. I'm not. Well, only if you use the microphone.
Hi.
Okay.
So I'm going to ask a question. So if somebody does this genetic testing and they find out they do have a gene and their child has the gene, what can their child do? Wait for the cancer, or are we looking at how to...
Yeah, that's a really difficult question. Really difficult question, because it's most advanced with respect to acute leukemias, acute myeloid leukemia. And when I'm teaching my students, I show them an example of an acute myeloid leukemia of a young woman who was feeling just a little bit off, and she went to a GP and got a blood count, and it was perfectly normal. And 2 weeks later, she was feeling rotten, and she went to another GP, got another blood count, and it was just florid, horrible acute leukemia. 2 weeks. So we can't really monitor patients to say you're about to get leukemia. Really difficult, and all that anxiety and stuff. So to answer that, to try and deal with that, there's a New Zealand consortium that's been set up. I've just forgotten the lead person. Just set up a few months ago, it was at the beginning of being set up, to try and come up with guidelines to say if you've got this particular variant or this particular variant or this particular variant, what do you say to the patients, and what sort of monitoring might be appropriate. Really difficult, because you don't want to spend a lifetime of anxiety, do you? Some people want to know, some don't. Thank you.